Good morning, all. Am I audible? Hello, am I audible? Today I am going to teach chromatography. So I have discussed what is chromatography, different phases of chromatography, concept of chromatography, application of chromatography in analysis of drugs, hormones, vitamins, qualitative, quantitative analysis, determination of molecular weight of proteins, separation of amino acids, proteins, carbohydrates. Types of chromatography: thin layer chromatography, gel chromatography, column chromatography, ion chromatography, gel filtration chromatography, gas liquid chromatography, affinity chromatography. Paper chromatography I have already discussed. Retention factor is discussed. The distance traveled by substance by distance traveled by solvent. Then thin layer chromatography has been discussed, in which a glass slide is used to separate the mixture of compounds. Then gas liquid chromatography is discussed, in which two phases are there, mobile phase and stationary phase, and passed through a column, and it is detected by a cylinder detector, and it is very rapid technique. Adsorption, in which adsorbent is used as adsorption layer, which is activated silica gel alumina, and serves as a stationary phase. In the mobile phase, will separate out the mixtures from the column, which can be eluted out. So this is column chromatography. It was advanced option for chromatography. Column chromatography, then ion exchange chromatography, anionic exchange, cationic exchange, two type of chromatography. Based on electrical charges, the separation takes place. Positive to negative, negative to positive. Differentiation is there. Two phases there are there, stationary and mobile phase, which are separated out. Then gel filtration chromatography. After ion exchange chromatography, there is gel filtration chromatography. It is based on the size exclusion chromatography, is also known as size exclusion gel permeation chromatography. It separates molecules according to their sizes, shapes, and molecular weight. It is referred as molecular sieving and molecular exclusion chromatography. Smaller molecules are able to enter the pores of the media. There is a bead which is made, and in that bead, smaller molecules are trapped. What happens when the smaller molecule passes through that bead? It is trapped in that bead and can't pass through that bead. So it is trapped. Then the bigger molecules can't be trapped to that bead. So it moves out, and its mobile phase is moving out and is collected and eluted out. But the smaller molecules remain in the pores, and it is eluted later. So it will move slowly. Packaging and student thus suffer essentially no retention. Uh, Such species are eluted first. This is how the molecules separate based on size. It is generally a low resolution chromatography. It is often referred to as final polishing step of purification, determining tertiary structure and quaternary structure. With differentiation of structures based on the sizes. So based on the different structures of protein can be separated. For example, tertiary structure and quaternary structure. Quaternary structure are larger structures. Tertiary are smaller. Can be purified. Proteins can be purified and separated out. This is the gel filtration pictography, in which the mixture is sample is placed, and the chromatography beads are there, gels are there, which will trap the molecule, smaller molecule, and let pass the bigger molecule. And the tubes are there to elute, which when the solute is eluted out, the mobile phase is eluted out, then it can be collected here.